Welcome to Careers in Discovery, your window into the world of biotech leaders. On each episode, we interview the industry's leading executives to understand how they built their careers, what they've learned along the way, and the advice they'd give to their younger selves. This week on Careers in Discovery, Emma is with Jordi Carreras-Puigvert, Chief Scientific Officer of Phenaros, a Swedish company at the intersection of AI and life sciences. Jordi shared generously how personal tragedy had inspired a career in science, his unconventional path through the industry, and the critical role that mentorship and networking have had in shaping his success. We also discussed with him the transformative potential of AI in drug development, and especially the importance of high-quality data.
Welcome back to Careers in Discovery. This week I'm joined by Jordi Carreras, Chief Scientific Officer and co-founder from Phenaros in Sweden. Welcome, Jordi.
Thank you very much, Emma.
It's a place to start. Let's talk a little bit about the work that you and Phenaros are doing at the moment.
Yes, absolutely. So we are a spin-out company from Uppsala University in Sweden. And we are a tech bio company, which means that we operate in basically the border between life sciences, or the intersection between life sciences and artificial intelligence. At the moment, what we are doing is we are developing our platform that is composed of a highly automated wet lab combined with artificial intelligence. And what we are doing is basically accelerating drug discovery through automation and through technology.
Yeah. And that's particularly within the rare disease space, is that right?
Yes. So we are, well, in a sense, our platform is both disease agnostic and also modality agnostic. However, we are focusing very much on the rare disease space because we think that there is a huge unmet medical need, and we think that our platform is actually very, very much tailored for rare diseases. The reason is because we work with high content microscopy using morphological profiling, and when we use patient cells, it's relatively easy for us to, for instance, combine healthy donor cells with patient cells, see how they are actually different, and then identify a potential disease phenotype, and then do very quick, very efficient drug repurposing screens, or also not with novel chemical entities, and then see how then we observe how these screens then provide us hits that reverse the disease phenotype. And this has been working really well. So we want to work on this space from at least for now.
Yeah, and huge impact can be made in that patient group, right?
Exactly, and this is definitely the main goal.
Yeah. So tell me, how did you decide on a career in science, particularly within drug discovery?
That's a great question that I really like to get, because I have an unusual sort of career path, and I will start from the beginning. So it all started when, unfortunately, my mother passed away when I was very young. So she died of cancer, and then from that point on, I think I was 13, and I really wanted to do science, to do something. So I set myself to study hard, and at the beginning, I wanted to have a very hands-on approach. So actually, I became a clinical laboratory technician. So I was really working in the lab, just analyzing basically the diagnostics. Although soon after that, after I started, I realized, well, you're not going to make it very far in terms of trying to come up with new therapeutics. So then I started looking for what I could study to develop further. And by then, that was in the early 2000s. Biotechnology was a field that was quite new where I come from, so close to Barcelona in Catalonia. And I decided to study that. It sounded very promising. It sounded like something that it was going to be revolutionary. Of course, now we're talking, 20 years later, biotechnology is you don't even need to talk about it, right? But at that time, it was an eye opener. So I was exposed to what biotech could do at many levels. You could go from production of pharmaceuticals to actually more molecular biology.
Mm-hmm.
I went that route, and that was great. After that, I realized, okay, so now I want to go further. So I want to do a PhD. So then I moved to the Netherlands, and I did my PhD in Leiden University. In there, I studied the DNA damage response. So at that time, we and many others, of course, we wanted to identify potential new targets, anti-cancer targets. So we focused very much on identifying new components of the DNA damage response. At that time already, I was starting to develop new technologies and new techniques, and we started setting up. Now it sounds really old, but we started setting up sRNA screens, large-scale sRNA screens, and that was very, very interesting and a huge learning experience for me. And then I also started working with microscopy, which has now become my passion as well.
Yeah.
So we started working with high throughput, high content microscopy, live cell imaging, which again back then it was pretty novel. And then after that, I moved to, so I finished my PhD. I did a postdoc working with C. elegans, which was completely different, but it was also very fun. And then I got the Marie Curie to move to Stockholm. So I met my wife. My wife to be, she's Swedish, so we moved to Sweden. Yeah. And at the so I was at the Karolinska Institute for 2 years doing a postdoc in the lab of Thomas Halliday. That was also a huge learning experience, because in that lab there was a lot of synthesis of new compounds, everything directed towards drug discovery. So during my PhD, we already realized, that you're looking for new targets, and what are we going to do with those targets? And then during my postdoc, it was very clear that identifying targets was one thing, and then drugging them was another thing. And it was a fantastic time in that lab. And then after that, I was recruited by another professor at Karolinska. His name is Oscar Fernández Capetillo. He has a dual affiliation in Madrid, in the Center for Cancer Research, and Karolinska. So then he recruited me to basically be the second in command in his lab. And that was a game changer for me, because I was basically building a phenotypic drug discovery lab from scratch. So that I did doing well, and then I basically co-run the lab with him for 5 years. Fantastic experience. It was absolutely phenomenal. And then I moved my research to Uppsala University together with Ulla Spjut. So his group was back then, 4 or 5 years ago, eminently computational. So the group had worked very much on building predictive models using structures, for instance, for safety assessment, and working with industry as well. So that was that was the main activity. But then when we met, we started talking, and he wanted to incorporate more cell-based assays to generate their own data, and that's where I came in. So I basically set up a second lab, a phenotypic drug discovery lab, but this time in Uppsala. So that's where I'm also now. So I'm associate professor and CSO of Phenaros. And then in the meantime, I also became the head of something called the Chemical Biology Consortium Sweden, the Uppsala node.
Okay.
So the Chemical Biology Consortium Sweden is a big entity that was created about 10 years ago. That it's basically a conglomerate of expertise in the chemical biology space. We offer sort of cell painting to Swedish researchers.
Right.
This is it's one extra activity. And then we formed Phenaros. So at Phenaros, I'm the chief scientific officer and co-founder. And what we're doing now is basically developing the discovery platform that we have put together. So that is, again, lots of automation on morphological profiling using cell painting combined with artificial intelligence. And what we're doing is we are, for instance, building predictive models for safety assessment. We're also running screens, as you said, for rare diseases. And this is now the main activity, and we are still a very young company, but we are growing fast. in a very nutshell, this is my career.
Very fascinating, and I think I take from that is how influential people have been, and that perhaps that mentorship aspect.
Yeah, absolutely.
What
So,
advice would you give to people to be able to take that opportunity for mentorship?
Absolutely. So I always say that I've been very lucky because I've been, already from my PhD, my supervisors were really keen on advising me also how, in what route to take, or they would just also tell me, well, go and explore, and see what actually you like to do. But also I've been very lucky on the other positions that I have had as well, because in terms of mentorship, which is really important when you're in early stages of career development. For instance, Oscar at Karolinska was a great mentee, or a great mentor. Sorry, and that has, I think it's very important. So if you are lucky to interact with people that are willing to guide you and to expose you to high quality science, to also make you see that there is also which are the possibilities within your field, or also allow you to explore other fields. I think this basically opens up your horizon, and you can see all the possibilities that you have in front of you. I think that also what is very, very important is to embrace those opportunities. So to be very, very active, not to just wait and see what happens. I think you have to put yourself out there as well.
Yeah, absolutely. That's great advice. Something that I'm keen to explore further is that the pivot from academia into the biotech environment. I often hear that there's a difference in freedom from a science perspective, and that there's more commercial drivers. What would you say to that?
Yeah, I think that so it really depends on what kind of academic environment you are. I also I've been very, very curious about the industry and biotech environment as well. I think back in the day, everybody had the idea or the conception that when you would go to industry, everything is production and everything is closed, and there is no freedom. And I actually think that I find it extremely fun to. Of course, I'm the co-founder. I'm one of the co-founders, so of course I have the freedom to decide what to do, right? But I find it. I find the structure of being in industry, and the way that, and the goal oriented approach, let's say, I think it's incredibly stimulating. And one of the reasons connected to this, why we also started the company, is because when you're in academia, you're doing, even if you're doing basic science or very advanced science that towards drug discovery, for instance, there is always the limitation that you will hit a point where you can just not do anything else. You find, you have a discovery, you discover a molecule, and what are you going to do with it? Sure, there are a handful of cases where that has maybe turned into a potential treatment.
Yeah.
But it takes time. It's extremely difficult. You have no funds. You need to start maybe connecting with somebody that actually can bring it forward, etc. But the number of hurdles is incredible.
Yeah.
While from being now in industry, we can clearly see that things move in a completely different world. So now all of a sudden, we have the opportunity to directly partner with companies that actually can facilitate that your discovery makes a huge impact. And this is a completely different story. And I think this is one of the main motivations. So we want to have in the world and for the patients, and I think that is a huge motivation for us.
Yeah. Are there skills and competencies that you've had to work on or to develop further for that move into industry?
Well, I have had to learn a lot about things that I had no idea about before. I mean, in terms of, when you're a co-founder, you start hearing lots of terms, for instance, in terms of financials that you had no idea about. Sure, there are some similarities when you get grants and things like this in academia, but it's just it's again another kind of terminology, another world. Yeah, exactly. It's like going up several ladders, I would say. This has been a steep learning curve very quickly. I think also learning the language of how how do you interact with a similar peer in a company, because in academia, I don't know, at least from myself and what I have learned and in the way that I have grown up scientifically, is to interact very, very transparently, very humbly and collegially with other academics, and in industry, I think it's still like this, but I think you need to. It's everybody has their own roadmap and objectives.
Yeah.
And I think at these kind of interactions, it's where you start learning. You need to learn a lot more about these kind of soft skills to interact with people at a different level. Yeah.
Yeah. So almost translating the science and the possibilities to meet the different objectives of the different stakeholders. Yeah.
Exactly. Yeah.
That's interesting. So, if you were to give a piece of advice to scientists that are early in their career, or perhaps people that are looking at making that move from academia into industry, what advice would you share?
The very first one is don't be afraid. I think that there is nothing to be scared of. I think that working in industry, and now we collaborate, and I know many people in industry. I think it's an amazing environment. So don't be. I would say, if I may, don't be afraid of going to the dark side because they have cookies. It just really it is a good, if you find a good opportunity, just try it as well. I think in early stages of our careers, we sometimes have the tendency to get stuck in a particular topic, or in a particular place, in a particular environment. And I think that it's very good to put yourself out there, to test new different things, and don't be afraid of maybe going to industry for a little while. You can always go back to academia. That's also an option. And I think that I would say, so when I talk to PhD students that are about to finish their PhDs and then trying to decide if they want to stay in academia or they want to go to industry, I always say the same: you have to be very, very active. So in order to find opportunities, you need to look for them. Yeah. So nobody's going to come to you and then offer you a position.
Those days have gone. Yeah.
Exactly. Yeah. Very good point. Those days are gone. Exactly. So you really need to put yourself out there, be active, interact. There is something that, I mean, now it's becoming a bit of a cliche, but this kind of network kind of concept. It is true. It can just, it can open many doors. I know that it's not easy for everybody to just, knock on a door and then say hi, but there are many ways nowadays to network and to find your space. And I think it's super important to put yourself out there. Yeah.
Okay. It's really good advice. Thank you.
Mm-hmm.
So what's next? What's next for Jordi? What's next for Phenaros?
Yes, I mean, it's super exciting at Phenaros. We are growing quite well. At the beginning, we were a bit slow at growing. Now I think things are happening a lot faster. So for Phenaros now, we are really establishing ourselves as a tech bio company that we are getting attention from big pharma, which is our objective.
Yes.
And we are working with them for accelerating drug discovery. And it's now very clear that the technologies that we're developing, they are very interesting for this kind of industry. And that's what we are focusing at the moment. So, and then in parallel, of course, we are also working with, for instance, interacting with patient organizations in the rare disease world, to see how we can contribute, and that is actually a very, very exciting. And what we are doing now is establishing our lab outside of academia, and and really growing the team to have the right expertise. We have a fantastic team at the moment, and we will be growing in the coming couple of months, which is very exciting. For me, what I see myself in Phenaros, of course, I mean it's my baby in a way. I mean, I have two real kids, but also this is one that you also need to take care of. And I very much enjoy working in Phenaros. I think we have a great team that is doing that. We're doing really, really good science and really fantastic things. So what I see is basically bringing the company even further, growing. And interacting with more pharma companies, with more biotech companies, with more patient organizations, and I'm really hoping that we will be able to make an impact, not only on rare disease in the rare disease space, but also making an impact on how drugs are more safely brought to market, and that's what we are focusing with a large pharma.
Yeah. And you touched on it earlier. We've seen AI make a huge impact into the speed and efficacy of drug development. Do you have any predictions of how AI will support further in the future?
Yeah, that's a great question and a very complicated question. I think that the AI, as you said, and it's very clear that it's evolving at a tremendous speed, and I think that there are plenty of examples where AI companies have actually facilitated the acceleration of drug discovery. I think that the field is moving very fast. I think that there will be probably new technologies, new models coming in very soon, actually, that will allow even better predictions, for instance, of targets and better predictions of molecules. I think that one thing that is very crucial and is very, very clear from now is that what we really need to do is to generate high quality data. Without generating high quality data and access to high quality data, then the AI will not really be as useful as we think. So, and this is actually something that we are focusing a lot in Phenaros. So we use morphological profiling to look at cells, how they change after we expose them to perturbations, and that is chemical perturbations or genetic perturbations. And there are some public repositories of this kind of images of cells, morphological profiling, But we realized that we needed better quality of the data that is out there. So that's what we are focusing on. So we are really proud to always say that we generate the highest quality data possible. We perfected the protocols, we perfected our microscopes. We actually have custom-made microscopes for cell painting and morphological profiling. And this, I think, is key. So to generate the highest quality data, because otherwise, even if we are the greatest AI lab in the world, again, it will not be as useful as people might think.
Yeah. Thank you for sharing that. I've loved having you on. It's been fascinating hearing your stories and your thoughts.
Thanks so much.
I'm wishing you and Phenaros all the best for the future.
Thanks so much.
Watch this space.
Yes, it's super exciting. So yes, thanks so much, Emma. It's been great sharing my story. I had a lot of fun. So thanks so much.
Thank you.
Thanks for listening. If you enjoyed the episode, please like and subscribe and share it with someone else you think will benefit. And for more podcasts, videos, blogs, and insights, come follow me, Tom Froggatt, on LinkedIn, YouTube, Instagram, Medium, or TikTok. See you next time.